Assalamualaikum and a very good afternoon. My name is Muhammad Fitri and I'm a master student from the program of Master in Analytical Chemistry in UPM, Faculty of Science. I am also a pharmacist working in National Cancer Institute in Putrajaya as a nuclear pharmacist. Today, I will be presenting a research proposal with title of uh, Molecular Dynamics and of molecular dynamics of PSMA radiopharmaceutical for cancer detections. Okay, here are my content outline for my presentation today. Okay, first, what is a radiopharmaceutical? It is a biologically active compound or molecule which has been labeled with the radioactive materials by which it can provide a physiological and morphological imaging of the patient. This can be done by injecting the patient with a radiopharmaceutical and scan them by using a machine called gamma camera, or well known as a PET-CT or SPECT-CT. Therefore, it is important in the development of the radiopharmaceutical to ensure that the biologically active compound will be properly bind to the correct and intended physical protein receptor. This is to aid the physician in planning and diagnosing the disease of the patient. All right, what is the prostate cancer? Prostate cancer is one of the most common metastasized cancer in men. As stated in the slide, it is one of the most common malignancy in men and having the highest cancer death among men. This cancer is derived from the prostate gland, which is a complex and heterogeneous disease. And it is by using the imaging techniques such as PET-CT, positron emission computed tomography, and computed tomography, it will give us an image needed by the physician to diagnose and stage cancer in a patient. Therefore, by using an antigen called a prostate-specific membrane antigen, we can use them to identify the specific stages of the cancer and could treat them by using a high-energy radionuclide for therapies such as beta or alpha emitting radiations. For example, uh, we are using lutetium-177, which have a beta energy that can be used in the treatment of metastatic castration-resistant prostate cancer. So what is actually a PSMA? PSMA is a molecular weight, a protein antigen with a molecular weight of 100 kDAs, and it consists of 750 amino acids, and it is a type 2 integral membrane glycoprotein. It has a unique structure containing 3 distinct parts. First is a 707 amino acid extracellular region, and cell membrane consists of 24 amino acids, and cytoplasmic tail, which contains 19 amino acids. This antigen has been used to diagnose the prostate cancer, and if the patient is suitable, we will use the mentioned radionuclides with which is a lutetium-177 to be labeled with the PSMA for a treatment called a targeted radionuclide therapy. The effectiveness of this treatment is shown on these figures. Next, I will show you a clinical trial. A clinical trial which was called a vision trial that has just undergone a phase 3 trial, in which this study has shown that by using a patient with lutetium-177 PSMA treatment has a prolonged overall survival by 15.3 months. We compared to the patient which only received a standard of care treatment. This this study has been done on 831 patients. This shows that by giving patient lutetium-177, it will be beneficial to the patients in the long run. There has been three most common type of radiopharmaceutical used by the nuclear medicine departments. First is the HBCC PSMA, which use HBCC chelators, but it is using for most used compound, but it's used mostly for imaging only because it is not suitable for a therapeutic radio labeling. And then PSMA 617, which use DOTA-derived chelators, which use mostly for therapies, and PSMA INT, which means eye energy and imaging and therapy, which use a DOTA-chelated urea base, can be used for therapy and diagnostic imaging. So this shows that not all of the developed radiopharmaceutical can be used for both diagnostic imaging and targeted radiotherapy. This is important because different compound will react differently. For example, when we use gallium-68 PSMA-11 for the diagnostic imaging and lutetium-177 PSMA-617 for radiotherapy, it shows a different side of attachment, even if it is the same PSMA compound. Therefore, it is important for us to know the property of this compound in order for us to facilitate the visualization of the metastatic lesion in the cancer patient. In this figure that I have mentioned before, like I explained, although there are studies like vision trials that have gone up to the phase 3 clinical trial, we still have unwanted results. So this means that we have not known any of the molecular behavior of the peptide towards the receptor. Therefore, by studying the molecular dynamics of the peptide receptor, we would have much more understanding of the peptide, and should we have more understanding, we can develop a much more better compound than these developed radiopharmaceutical peptides. For the reason I mentioned before, I would like to do a study on molecular dynamics and docking simulation for the PSMA peptide with the receptor in order to understand more on the relationship between peptide receptor complex. This study will have the objective of the study of molecular interaction between the peptide and receptor using molecular dockings, studying of the dynamic of peptide receptor complex by using molecular dynamics simulation to analyze the mechanism of the peptide by mechanism of peptide binding to the receptor. Okay, so this is my methodology. This is all I will be explaining in the coming slides. For the preparation of the peptide, such as PSMA protein and the peptides, I will download a PDB file from the PDB data bank, and the code for the protein of the prostate specific membrane antigen is 1Z8L, which is a crystalline structure shown in the figure. It is prostate specific membrane antigen, a tumor marker, and a peptidase. The protein will be processed by using Yasara software. As for the peptides, I will download the STF file from the PubChem and run it on the Marvin sketch, thus process it using the Yasara for the docking simulations. After the preparation, I will prepare the parameters for the docking simulation using Yasara software. And then I set the force field, simulation condition, and simulation box. After the docking simulation and the data has been obtained from the Yasara, the data will be analyzed by root mean square deviation for biomolecular structure evaluation, flexibility of the compound by using root mean square fluctuation, evaluate structure change during protein folding using solvent accessible surface area. From this, we will know the interaction between peptide receptor during the docking simulations. After that, molecular dynamic simulation will perform and all the parameters accordingly will be set by using the SARA software. After the simulation ended and the data retrieved from the YASARA, the result will be analyzed by using will be analyzed as the docking data. From the result, we hope to analyze the molecular behavior of the peptide receptor complex and measure how much the different protein conformation has changed over the simulation conditions. From all of this, we hope to understand the behavior of the protein to the receptor. These are my project management, my reference, and thank you.